seminar. So it is my pleasure to introduce you today Professor Weilong Hao from our neighbor institution, Wayne State University. So Weilong is a molecular evolutionist who actually has worked on quite a diversity of organisms, from bacteria to fungi to plants or their mitochondrial genomes. So he's a molecular evolutionist with a specialty in bioinformatics and comparative genomics. And so his major foci of the research are horizontal gene transfers and homologous recombination. And then mitochondrial genome evolution and mutation, which is the topic he will talk to you about. In addition, in the past, he's worked on mitochondrial nuclear interaction, and especially in the plant system, when he was a postdoc with Jeff Palmer at Indiana University, who used to be here. And lastly, he worked on comparative metagenomics and dynamics microbiota. So Weilong got his undergrad degree from Nankai University in China, which is a leading higher education institution over there. So his degree was in microbiology. Then he did his PhD on the other side of the border, McMaster University in Canada. Then he did his postdoc in Indiana University, Bloomington. So 2012, and he joined faculty at Wayne State. So today it's a pleasure to have a young fellow colleague from there. So without further ado,
all right, yeah, thank you so much, Vacu, for the invitation and the introduction. So when I, the first got the email from Vacu, I mentioned this to my wife, said, Vacu invited me to give a talk at the UM. My wife said the first sentence was that you better have something smart to say there because, a bunch of smart crowd there. I'm like, well, unfortunately, I don't think I have anything smart, but I just hope that, that I tell you something like, you have not, been fully aware of it for some time. I hope that's the case. Let's see, how it goes, okay? So I'm going to talk about genome variation and mitochondrial evolution in the yeast system. But this is, since it's EEB crowd, I don't think I need to introduce you anything about mitochondrial genome or yeast, since we have really several lead labs working on yeast, and we study evolution. We know, the mitochondrial genome, right? But some of you may not fully aware is that how diverse or how different those mitochondrial genomes are. If you think about this, if you look, if you are, interested in, Homo sapien, which, we have a number of them here. So the genome size is like, 16 kb, okay? If you care about, say, Drosophila monogaster, the mitochondrial genome is like, 20 kb, right? If you care about, Saccharomyces cerevisiae, but in this particular strain, they sequenced, they show you the, the genome size is about 80 kb. Again, even Saccharomyces cerevisiae, the variation is still huge. Now, if you check the gene bank, the biggest is probably close to like 100 kb. The smallest probably is smaller than this. It's like 70 kb. Okay, so now if you really care about umbrella, the Nicotiana, the tobacco, so you see that actually the plant mitochondrial genomes are a little bigger than that. They're like 400 kb, and there are even bigger ones I could not put here. One plant species called Salini. I think the mitochondrial genome is like the 10 or megabases or bigger than that. It's like bigger than most of the bacterial genomes. Okay, so we are interested in the mitochondrial genomes in different ways. But before I start my own lab, I never worked on yeast before. Let me tell you, this is more like how my research evolved, from the very beginning, the first, the study we actually conducted, on the yeast mitochondrial genome. So again, when you had no experience on mitochondrial genome or yeast mitochondrial genome, you want to make comparison, right? If you, to look at the human genome, if you look at the yeast genome, I don't know about you, when I first look at this, I noticed two things. One is that the genome size are different. I'm sure everybody can pick that, right? Another thing is that even though a small thing you may pick is that those NAT genes are missing from yeast. If you talk to Tim, and the team may also tell you, in many other or several other, fungal species, they also don't have the NAT genes. But those are not the real thing I first pick up. I picked up something like those. If you look at those bars, those are introns, which in humans we don't have. Okay, they're in Saccharomyces cerevisiae, you have 3 genes. Most often they have introns. You may say, okay, who cares? Introns, we don't care. But let me tell you, actually, it turned out actually it's very interesting actually to look at them. So, give you a little bit background. If you ever, to look at, the papers, say plant mitochondrial genome, they may also say that actually, in those genomes, you have those introns. We call it a lot of times the group 1 intron. Those are one of the ancient forms. So a lot of them in the exon region between them, you have this intron, but also you have an open reading frame. Those open reading frames encode endonuclease. So basically, within the intron, there is a gene. This gene has a function. The function is likely in the nucleus to cut the DNA. Turned out that the little protein help the introns splice out and move around. That's why they call it mobile introns. So when we look at, again, this is when Baojun look at it, they say, okay, there are a lot of, in this particular gene, is that, what he first look at is this 21S because it's simple because there's only one intron. If you look at the COB gene or COX1, there are many. It's so hard. We just start something very, very simple, right? Let's go back from the very simple case. So when he looked at this, he told me, oh wow, I found, because if you look at those axes, that means in that particular gene, the intron is missing. Okay, you have no intron. So basically, just the 21S ribosome RNA, that's it, perfect, very good. Some of them, you have the intron, not the open reading frame or the HEG, the endonuclease gene. So he showed me this. So basically, map them on the tree. I'm like, yeah. So there are a lot of differences. So what, right? So basically, again, if you are, the student or postdoc, talk to your PI, the PI didn't show that much interest. Well, there are two things can happen. You may say, okay, it's not cool. I'm not going to work on this. Another way is that I'm going to try to make it cooler than you think. So actually, that's what he did. He's trying hard to think about this. He actually, because he's very detailed, pay attention and show me, okay, this is the sequence alignment. Again, if you think about the sequence alignment itself, it's very simple to generate. It's very easy, make alignment, show that, right? You show, okay, he showed me, and also look at the literature. So the intron boundary, the exon sequence, also there the recognition site of the HEG, or basically the endonuclease also recognize this place and cut, insert the intron. Makes it very convenient. The intron can move around easily and cut, insert, cut it out. And he showed me, okay, even within Saccharomyces cerevisiae, this is the same species, 3 strains he looked at, plus, minus. That means within the same species, you can have some of them have the intron, some of them don't. Even this by itself, you don't see, even in the nuclear genomes included, you don't see this often, or period, you don't see this period. Within the same species, some of the individuals have a lot of this intron, some of them they don't. You don't see this very often, okay? And he said, okay, that's kind of cool, but it's present, absent in the sense. But he said, well, actually there's something else. If you look at this scale, you have the exon region at the ends, you have the intron here, you have the HEG in the middle. He showed me the sequence, say, let's look at this carefully. Let's just look at the intron region first, the second. So basically, you have the intron region, so which is here and here, okay, very close to the flanking of this HEG. I'm convinced, if you look at the grade, so in those species, those are very different yeast. This is called Pterospora. Those sequences are actually identical with some of the Saccharomyces cerevisiae intron. But look at the third. It's a close related to Saccharomyces cerevisiae, has very different intron sequence. Okay, I think, I hope you, but on the right, the same sequence actually is identical to something else. Then also, if you look at in between the HEG, some of them have the HEG, some of them don't. It seems like the HEG, there is presence, absence, even within very close related species. And the flanking region may show very similar sequence with some distant related species. If you think about it, this is more like some bacteria, even we all call E. coli, some of them, they have the antibiotic resistance, some of them they don't, right? If you think about that, whether you call them horizontal gene transfer or you call them mitochondrial recombination, but whatever term you like to use, I'm convinced that actually those sequences have been exchanged in some form, whatever you call them, whatever you name them. So this is not enough. He also showed me that even within HEG, okay, within this open reading frame, some of the regions you see very similar to some distant related ones, but some other regions may be closely related with something else. That means even within the same gene, endonuclease gene, you can have some regions recombined with some other things, some of them recombined with some other things. Okay, it's like you can have recombination take place literally everywhere, within the gene, within the intron, within the HEG. So this is basically what we actually believe by looking at this very simple analysis. So again, this is not enough. As I told you, Baojing is a very careful person. I guess he look at when he generated the sequence, he's trying because he wasn't because I was pushing him to say, well, I don't believe this, or I don't feel it's exciting enough, and he's trying to get more stuff, interesting stuff to me. And this is something like he look at like this is it's another. This is more like a partial sequence from the full alignment. I showed you here, this is one of the regions I showed you earlier, but something I didn't really pay much attention is this. He actually showed me, look, this is bizarre. There is a small insertion, and this insertion is really GC rich. But if you don't study the mitochondrial genome, you may not fully aware is that the yeast mitochondrial genome, they are super AT rich. So say Saccharomyces cerevisiae, the GC content is 17%. If you think about E. coli, it's like 50-50, right? 50%. The mitochondrial genome in general is AT rich, even like 20-something GC content or in the teens. But if you look at this small region, the GC content is like close to 70. So really GC rich. And actually, again, I didn't know much about this. Actually, he tried so hard to look for some kind of interesting thing. He looked at the literature, and he found a lot of excellent studies by some pioneer, the yeast, the mitochondrial, the people. Actually, they characterize that in the yeast mitochondrial genome, there are fungal mitochondrial genomes that in general that you have this kind of like, by, the hairpin structure. People even hypothesize them, can be ribozymes. So that means more like the group 1 intron, they move around. So when you actually make a RNA structure, this is not exactly this one. The reason I show this is that this is the most popular GC cluster in the Saccharomyces cerevisiae mitochondrial genome. And then, I talked about, so let's look at this. So what's going on? Is there anything, interesting there? He actually collected a number of Saccharomyces cerevisiae genomes, actually, checked basically the whole genome. Okay, do you have this particular GC cluster or not along the genome? Luckily that the mitochondrial genome, if you align them, they're literally collinear. You have big chunk of insertion deletion, but they don't have a big rearrangement, if you like. So that it's actually reasonably easy actually to look at them. And you first thing I get, I hope you will get too, is that if you look at that, those GC clusters, you have very fast or dynamic turnaround from presence or absence or insertion deletion, if you like, whatever term you like to use, right? You see that the very close related one, some of them may share a lot of them, but also they differ some. If you look at, really depends on which one you look. So this convinced me, at least, is that those ones actually they are very mobile or dynamic. If you put this the same way as, if you think about this the same way as we think about the introns, is that the introns actually have the presence absence, but the recombination can facilitate the presence or absence or this kind of dynamic. This actually, we think probably is the same, that the GC clusters, they mobile because people think about they are the ribosomes, but we also some evidence that the flanking region, they show very similar pattern as the intron, the flanking regions. So then it made us think that those GC clusters, they're more like the introns. They're mobile, maybe, and they're facilitated by the recombination. Again, those are just cases, right? If you really want to convince people, especially at the time, when people did not appreciate the mitochondrial recombination as much as today, because even today, because the couple years ago, because this the Drosophila study, done by the Macro-O'Farriss lab, they actually convincingly demonstrate in Drosophila you actually can have very frequent mitochondrial recombination. Then the people start to believe that, but this was before that. One way to do that is that in order for me to believe mitochondrial recombination is convincing, is that I need to have some really close related genomes to compare. So if say that close related ones, you see differences, and the differences is very similar to very distant related ones, you then have the power to say, right? Okay, they're real. In order to do that, we actually generated the genome sequences from multiple genome sequences from the same species. Say Saccharomyces cerevisiae, there are several of them already by then. So we generated one Saccharomyces paradoxus. We generated a bunch of Pterospora. So and those Linchinsia, many of them were available by then. So basically, we have three major groups. So within each group, you have multiple genomes are close related. If anything, if there are recombination going on, you should be able to say those recombination are not phylogenetic artifact in a sense. That's how actually we designed the experiment. We want to test it that way. We have the mitochondrial genome fully sequenced. We only use the ones we can put up as a full circle. If they're not complete, we actually, there are many more we sequenced that we did not include, in this study. Again, just like what we did before, we actually check the introns, right? Because the introns, there are not that many. You can count by fingers. It's very easy to actually to check. But if you see that the black and the gray, because the black introns are the introns, because there are a number of very excellent studies have done that experimentally to show those introns are mobile or not in Saccharomyces cerevisiae. So those ones are the people think active ones. The gray ones, they are the ones people think not as active. But one thing you can see that whether the black or gray, you see that within the same species, different strains may have different intron patterns. So this actually, there is only one exception probably here, because these two strains, what we sequenced, we believe are the same strain, because even though they call the same strain, the different strain number, they differ by just one nucleotide. Again, we believe they are not even real the two different strains. But other than that, all the species actually we have, if they have multiple strains, we didn't even sample that many for each species. We see that there are very different intron patterns among them. So if you think that's not convincing, actually we made tree for all the introns have certain number, I think 9 sequences that we actually can put. For each one of them, we actually can have for each one of them, we actually can see that. I just talk about the one as the example that if you look at the color, the three colors are the three groups I talked about. If you look at the dots, so if they're the same color, that means they're the same species. Okay, that means the 3 Saccharomyces cerevisiae, or 4 in this case, even though they're the same species, they're not even together here. Okay, if you look at here, this Pterospora, the Tabruki, the Bruki. So actually, they're the Pitoriaceae. So they are not even together, even though they are the same species. If you look at all others, they show you literally the same story. Okay, all those introns, they have been exchanged in some way. So we also look at within the intron. So like one part, okay, look at those guys. The beginning of that intron, these 3 are together, these 2 are here, okay. But at the end, actually, they are together. That means the recombination happened within the intron. It's really hard to make the argument. Those introns, they cut out, they move around, inserted into another species. That means you have to have recombination going on right in the middle of the intron in this case. Okay, that means recombination happened. It can happen even within the intron. It can also happen within the coding region. So this is the COB gene. If you look at the first part, if you look at the second part, you see that here actually how they clustered and how they cluster here. Basically, the one gene, one part they show one phylogeny, the other part show a different type of phylogeny. The recombination happened in middle of the coding sequence. That means again, a lot of times we don't see that if you actually sample very different species each time. Each species, you just sample one strain. Of course, a lot of times you don't get this kind of resolution. You don't get this. So here, actually, we are convinced that the mitochondrial recombination can happen very frequently. It's not like they're rare. They're actually frequent. The reason is that why? That's funny. It's flipped. It should be upside down. In this case, but anyhow. So why they are frequent? What we believe, is that because the yeast, we all know that their alpha mating type, A mating type, they actually mate. If everything goes well, they will fuse their nuclei, basically, from the haploid, the single cell merged into the diploid organism. But we never think, or we rarely think about if things don't go well. They actually will separate, right? They actually, they will separate into 2 cells. But if that's the case, because of this, the mitochondrial fusion, even the mating is not successful, the mitochondrial DNA may still get the chance to exchange. So you can actually create, this kind of recombined version of mitochondrial DNA. So in order to actually test this a little bit further, in a way that we ask a very simple question. Let's identify some species people don't think they recombine, or they don't think they mate in the lab, which in this case is the Candida glabrata, because at Wayne State, actually the dean of the medical school runs a women's clinic. You have the Candida glabrata, you actually can collect from the patients, and also Saccharomyces cerevisiae is also a pathogen. Actually they actually have the Saccharomyces cerevisiae and the Candida glabrata from the same region, from Detroit, and from I think the same 3-year time period. So basically, we are running this, what we think is a little bit controlled, better controlled, not the best way, but a little controlled experiment. We have 2 species. They're all sampled in the same city in the same time period. We actually can compare this. We have 30 Saccharomyces cerevisiae, 30 Candida glabrata. But it turned out we didn't get all 30 because sometimes this one didn't grow that well, but we prepared the sequencing library. Some of them failed, but overall we got almost all of them close. But the one thing, quick 30, the data I show you is here. You can first look at the intron pattern. That's what I showed you before. And also you look at the intron pattern for Candida glabrata. So you notice this Candida glabrata, there are only 4 introns. Okay, but this is not what I really want you to look at. What I really want you to look at is that this here, one intron, these 4, they don't have this intron present, but these 4 strands, they cluster together. Okay, but this intron is missing in all those guys, but those guys cluster together. You can easily say, okay, maybe just an intron, gain or loss event. You can use that to explain the whole picture. But if you look at the Saccharomyces cerevisiae, especially the ones actually I put, the asterisk there, you just cannot use those to explain, because those trees are the mitochondrial gene tree. So basically, those are the evolutionary history of those mitochondrial genomes. You have to have some kind of mitochondrial recombination or exchange in order to create those guys. The close related ones have very different intron pattern. You have to have that. That means by comparing these 2 species, one actually can have sexual reproduction, the other one in the lab we don't see mating, we could not even make them mate in the lab. You actually do see a significantly reduced mitochondrial recombination pattern. So we believe that the mating or the unsuccessful mating can still facilitate mitochondrial recombination in yeast, even though they are not successful, but the mitochondrial DNA actually get the chance to exchange their DNA. So this is actually what back the belief we had. We believe the mitochondrial recombination in yeast actually is very frequent, but the reason we often could not see is just like what we did in the experiment. Let me show you is that if you have, say, there is a intron containing exon, when they recombine, you actually gain a whole intron or you lose a whole intron, you can easily detect, right, by whatever sequencing method or whatever way, probe. But if you have 2 sequences, they have roughly the same length, the differences, nucleotide differences, not much. You just cannot easily tell those several nucleotides, are they mutation hotspots or they are truly because of the recombination. So it's really hard to tell that way. So we think that actually the indels can help us to detect. That's the reason that we detect a lot of horizontal gene transfer cases or recombination, is because that in the yeast mitochondrial genome, there are a lot of indel mutations in the first place. So let me remind you that we talk about the mitochondrial genomes as very different. Since we sequenced a number of them, a good number of them, we actually can ask the very simple question, right? So how different those mitochondrial genomes are among those species. So actually we mapped the genome size on the tree. You have those terminal branches. For some, they have multiple strands. We just take the average. You notice that Saccharomyces cerevisiae, the number also changed, from time to time, when I talk to you. It's because this is the average. We also estimated the ancestral stage. If you notice that I will use the color, that if you see a red, that means from the ancestral stage to this the extended the node, you have an increase of genome size. If you see blue, there is a decrease in genome size. If you have black, the difference is not as much. The point I want to tell is that if you look at, you have different colors. You have also, the color that happen here and there. That means the genome size can change very quickly along on this tree. And also that you see that, some of the really big genomes can be close related with some really small ones, right? Some, tiny ones can be, close related with some, the decent sized ones. The genome size change very quickly. And we want to ask, again, very simple question. So why? What made this happen? We look at certain things people usually think. For example, introns, right? If you have a lot of introns, I expect you have bigger genome. If you don't have a lot of introns, of course, that was my naive assumption. We look at, okay, the intron length or their GC cluster we talk about, tandem repeats, or the homopolymer repeats. Again, those for eukaryotes, there are a lot of those. we look at the variance, of course, that here you see that the tandem repeats actually they have a higher variable. The biggest one, had a lot of tandem repeats that had a lot of variation. The introns, there are also a fair number of variation. We actually make a test to ask the correlation. Do you have the bigger genome associated with more intron size? Actually, surprisingly, the intron size or the intron length, they don't show a positive correlation with the genome size. Okay. However, if you look at the tandem repeats and also the GC clusters, actually there is a positive correlation. That means the bigger genome you have, but this is the contribution in terms of the fraction. It's not only they have more GC content in length, is they have more GC content in fraction. So basically I'm asking that is that if you have more intron, so that means if you have 10 kb in 100 kb sequence, that's 10%. If you have 20 kb, 20, that's also 10%, the same. But I'm asking, do you see a positive correlation of the fraction? In this case, the tandem repeats, the bigger one, they do have higher fraction of tandem repeat. So since the tandem repeats GC cluster, we want to ask, do they have higher GC content or lower GC content? At least we know that the GC cluster, they are GC rich, right? So we actually plot the genome size versus the GC content in the mitochondrial genome. We see a very strong negative correlation. That means the bigger genome, they have more AT-rich genome in the yeast. Then we actually compare them very carefully. We learn that those are the 3 genome pairs. If you look at the tandem repeats in all 3 pairs, they are super AT-rich. That actually backed what we found, that because in the bigger genome there are a lot of tandem repeats, because those tandem repeats are AT-rich. So those AT-richness really add the length and also reduce the GC content in this case. And we also discover something what we think interesting, but we don't have a the genetic mechanism to explain, is that the homopolymer sequence, you actually have a much higher GC content. That means in the yeast mitochondrial genome, the AT sequence, the proliferation, they tend to have tandem repeat proliferation. But the homopolymer sequence, their GC homopolymer sequence tend to actually proliferate in that sense. For that, the homopolymer, we don't have a really good genetic mechanism. But for the tandem repeats, it's very easy to understand, because the AT, there are 2 covalent bonds versus 3 is easier to have slippage happen. Since we actually calculate the GC content, why not, right? Compare all of them and look at the most AT-rich ones and see what's going on there. Actually, if you look at the intergenic region, the Candida glabrata is pretty AT-rich. But there's another one, actually, it's even more so. This is the guy. Actually, we want to look at it even further. If you look at the whole genome, you see that where you see a peak in terms of GC content, that's where you expect a gene most times. And other than that, the intergenic region is pretty flat. That means the GC content is very, very low. It's almost like, it's close to 0 even, very, very low. So we want to ask why. Okay, we know that if you study mutation, you know that there is a mutation bias towards to AT, right? Is this because of the fast mutation or elevated mutation towards to AT? We actually made a tree, this compare the sum, not all the yeast that we actually look at it. So this is the most AT-rich ones. The bar is the genome size, but those are the tDNA, the CDS, introns, if you pay attention, you notice that the average, the whole genome average, often is lower than the tDNA, lower than the protein coding, even lower than the intron. So basically, the intergenic region in those yeast, they actually made all the AT richness. And also, if you pay another attention that here, this bar is actually, that means this species, the Harsinia spora, Uvara, so actually had much faster evolutionary rate than this AT-rich, super AT-rich genome. That means the elevated rate is not the explanation for the extreme AT richness in this particular species. So then what else? We actually paid a little bit more attention, played a little bit the calculation on this. We know that the AT richness is really high. We want to ask what's the expected AA pattern. That's like dinucleotide frequency, AT or TA or TT. You actually can look at it. The expected, these are the dots, the observed, those are the bars. You notice that AT or TA, they have higher than expected frequency than AA or TA. If you recall that the homopolymer mutation, I would say that very likely they are deleterious. They are under selection. You don't want them, and you have AT or TA, the frequency. But if you look at the tetranucleotide frequency, the biggest bar you have, this is AT-AT. This is the TA-TA. So these actually fit pretty well as what we talk about the tandem repeat, right? You have a TA, you have a TA, you have a TA, you proliferate. So actually those frequencies are more than expected. Again, if you look at the underrepresented, those are AAAA, and this is AAAT. That's also increased the homopolymer run. So basically, what in this super AT rich genome, you have a underrepresented homopolymer run in this, but you have a overrepresented, more like tandem repeat in this kind of signature. Since we only did the tetranucleotide analysis, we didn't even go further to check others. So I hope by now you look at all the AT richness. So we develop a sense that actually the mutation in a way that has certain kind of pattern, they actually go by the sequence or sequence dependent. We want to use the mutation accumulation, try to understand, because we had this kind of feeling that if the mutation accumulation can tell us that would be really great. So since Mac Lynch offered that they actually sequence the wild strains, if you do the mutation accumulation, we have a lot of undergrads at Wayne State actually they do this for us for free basically, and also take the credit yeah, let's do this. So we picked these super AT rich ones and some close related ones that we actually did the mutation experiments and the mutation accumulation, and we want to ask, we hoped we can get some mitochondrial mutation data. We expect that we will have some nuclear mutation data, but we are not going to talk in detail today. But this is what we had for the nuclear mutation. We had a very nice story there, and actually the faster mutation, they're also consistent with some kind of increased oxidative damage. The story is is faster growing, it's all good. But what if you look at the mitochondrial data? That was actually a little bit to me disappointing, because the error bars are big, so it was not. But I think we still learned quite a lot if you pay attention to this bar, even though it's big, but they are the same. This is 10 to the power minus 10. This is 10 to the power minus 10. That means the mitochondrial mutation rate in this particular strain is still much higher than the nuclear in this particular strain. That means in some yeast species, the mitochondrial mutation rate is higher than the nuclear mutation rate. But in some others, it's not as much as dramatic, as we talk about it. If you pay attention to the slide earlier, you see that this branch is also the longest branch, faster evolving. This is fourfold because it's the sick bar, because I did not show the scale. This is 4 times per unit longer than that. So again, there are mutation variation, mutation rate variation there. You actually can measure that, in the sense. So since days, we actually developed some kind of ability, right, to do the mutation accumulation experiment actually to analyze that ourselves. We actually picked 3 strains. Basically, in the yeast, you can get rid of the mitochondrial DNA easily, because one guy we call the root 0. This is also the petites that people usually use, or you see on the petri dish. Because Also, you can actually replace the mitochondrial DNA easily. You have all of those. They have the same nuclear background, Saccharomyces cerevisiae, but this guy has the Saccharomyces cerevisiae mitochondrial DNA, but this guy had Saccharomyces paradoxis mitochondrial DNA. So let's do the mutation accumulation and see what happens. Okay. So this is what before that or during this, we measure the growth of the row 0, the native mitochondrial DNA strain, the guy with or the strain with foreign mitochondrial DNA. We notice something very striking here. Basically, the petits or the row 0 grows faster because this is the generation time or the time for cell division on this particular media, right? So actually, this one grows faster than that. So basically, the cell or the strain with the foreign mitochondrial DNA grows actually slower than the one with no mitochondrial DNA. So from there, it seems like there is a mitochondrial genome, nuclear genome incompatibility issue, but it grow. We did grow this, try to grow this on glycerol. It grow. It grow the fine. Looks fine. But it's still, if you measure the cell division in terms of the time, actually grows slower than the row 0. Okay. But if you compare the nuclear mutation rate, the nuclear genome, actually this row 0 had a faster nuclear mutation rate. If you think about that, this is a very, with no functional mitochondrial, it's a very stressful environment, and also the mutation spectrum with increased G to T change, which is the signal for oxidative stress, you actually can see that. It's in part, I think that explains the why the row 0 actually had higher nuclear mutation rate. But again, it's kind of surprising, even though the error bars are big, is that the foreign mitochondrial DNA did not show higher mutation rate than basically the native mitochondrial DNA. So what we think is that because the different mitochondrial DNA, they're also very different in terms of their sequence, the GC content or the number of GC clusters. We think in here, the differences cannot be explained by incompatibility because, if you look, if anything, this one should have, foreign mitochondrial DNA should have higher mutation rate, but, we didn't see that. Even though the trend actually is the opposite, but the error bar is big, we cannot say anything for sure. Right? But again, we think here is that the mitochondrial DNA, maybe the sequence also play a role, in terms of the mutation rate. The function itself, may not be, as important, in this case. Let's recap. I hope, I told you something that, the mitochondrial sequence, you have the presence, absence. Actually, some of the sequence, for example, the intron, the GC cluster, those sequences can be mobile. They move around. But they are greatly facilitated by recombination because recombination can quickly spread or delete a certain kind of sequence, so we can notice that. They also can use as a marker for us to detect mitochondrial recombination. So if you think about, the yin-down mutation, in my opinion, is that, the substitution mutation cannot increase or decrease your genome size by themselves, but the yin-down mutation can quickly increase or quickly decrease the genome size. The yin-down mutation actually, should be considered a very predominant force, in the mitochondrial recombination, and actually is sequence dependent in terms of the different mutation pattern we have. Also, we think, again, we don't have a strong proof for this, is that the mitochondrial DNA can impact the mitochondrial mutation, but we do have some kind of proof for the mitochondrial DNA impact the mutation of the nuclear DNA. I will stop here. Well, I do have some, people acknowledge, the people acknowledged on the way. There are some undergrads, to put it underlined there, actually, did a lot of the mutation accumulation study, and some of them, they did. I did not have space to put them on there. So the Candida glabrata, actually we got from, Jack Sobel. The mutation accumulation, the stuff we didn't talk about, but we did learn quite a lot, from Mac Lynch and, the Max Firmer postdoc Hong An right now is at Asian University in China. Other than that, I will take questions and thank you for your attention.
Thank you. That's a very interesting.
Yes. Well, yes, I believe so. But what I didn't talk about is that because those HEG, they move around also. It's, if you compare, say, different Saccharomyces cerevisiae, some of them, they have HEG, some of them they don't. So what we believe, because some of them, I cannot say all of them, some of them, the HEG still have function. So they actually, they move around, they actually facilitate, if they are in the nucleus, they should, do something, right? That's what I believe. But again, I don't have very, strong data to demonstrate that. I would believe HEG actually, to help, the mitochondrial recombination in a way. Mm-hmm. Mm-hmm. After cell fusion, they may get separated, but their nuclei never fused. Well, yes. Well, if you think about it, when we talk about, their prozygotic isolation, there are also post-zygotic isolation, right? If you think, okay, pre-zygotic isolation can separate certain type of species, in my opinion, should be very distant related ones. The close related ones, they may suffer post-zygotic isolation. They may can still fuse their cell, but that will still create a lot of opportunity for the mitochondrial genome to exchange. That's at least that's what our hypothesis works. There are papers demonstrating the cells can fuse their cell. They don't fuse their nuclei, then separated. There are actually pictures on that. There are reports on that. Yes. Yes. Mm-hmm. Well, you ask the expert there. So I would say that you have, even I think the Paul Nigalski even proposed there are even the zygotic isolation between Saccharomyces cerevisiae and Saccharomyces paradoxus. Even between these sister species, we actually can even make them mate. So again, post-zygotic isolation between these sister species, we don't see very strong. But again, some people demonstrate when they replace the mitochondrial DNA, even though they have the same intron pattern, but some of the introns may not get spliced out properly. There are a lot of things going on. It's very probably strain specific even. But if I don't know which one is more predominant in the wild, but you definitely expect both to happen in certain kind of degree. But again, in this case, regarding the mitochondrial exchange, as long as those cells fuse their nuclear, fuse the cell, they don't fuse their nuclei, they will provide the chance at least for the mitochondrial DNA to have exchange. But I don't know, you don't really need the post-zygotic isolation. As long as they get separated, they have the chance. I would say that the mitochondrial genome may have a higher chance to exchange between species than the nuclear genome, if you think that way.
Other questions? Okay, if not, let's thank you one more time.
All right, thank you.